Good evening. How are you doing tonight? I am Asiya Ermukhambetova. I am a scientist, a chemical engineer, and your host today. Welcome to Science Stars Kazakhstan. And before we start, thank you for all coming. And before we start, let me remind you on safety. There is no emergency training planned for today. So in case if you hear fire alarm, please leave the room quietly and quickly using this exit door and turn to the left, okay? And please, can I also ask you to switch off your mobile phones or turn them to the mute mode? Thank you. Today we are hosting our Science Stars winners and finalists of FameLab, an international science communications competition. Brightest young scientists from Kazakhstan, Spain, Norway, Malaysia, and Northern Ireland will present their ideas and tell stories to you. The event is organized by the British Council with the support of Chevron and by Nazarbayev University and held within the framework of Astana Expo 2017. Please welcome Jim Battery, who will tell more about the event and explain some rules to us as well. He is a UK Attaché for Culture and Education and Director, British Council, Kazakhstan.
Thank you, Asiya. Good evening, everyone, and welcome to Science Stars. So, as Asiya said, tonight's event is part of the UK's programme for Expo. But what part? Is it part of the education programme, or is it part of the cultural programme? Well, it's both, isn't it? Because tonight is where art meets science. And tonight we also are exposing our participants, our competitors, to share with us one of the oldest art forms known to mankind, the art of storytelling, telling a good story. You'll all know that the art of a good story requires you to capture your audience's attention very quickly and hold it and fire up their imagination. Our contestants tonight have 3 minutes to do that, to take us on a journey whereby those of us who are non-scientists can actually understand some of the latest breakthroughs or the more complex scientific concepts. Are they up for the challenge? You bet. They have done this many times before, to great effect, internationally. But a competition like this doesn't just happen alone. Not only does it require years of studying, hours of practicing, but it also requires good partners. And tonight's event would not be possible without good partners. I would like to thank our esteemed judges, you'll hear more of later. I'd like to thank Chevron, Nazarbayev University, and of course, the Cheltenham Science Festival. Without Cheltenham, we would not have been exposed to FameLab, and tonight's competition is inspired by FameLab, probably the world's most popular and best-known international science communication competition. That's all I want to say for now, because the people you really want to hear from are our young scientists. And it surprises me, you know, that whenever we have these kind of competitions, it's always the darker side of humanity that our scientists focus on. Sex, drugs, and rock and roll. Always those kinds of subjects. But I think that might do the trick and capture our attention. So good luck, everyone, tonight. Thank you, judges and audience. Hope you enjoy. Thank you.
Thank you very much, Tim. I'm going to introduce judges. Please welcome our judges. Janar Kushpanova, Community Engagement Coordinator in Chevron Company. Abdildar Shamenov, President of the Alliance of Technology Commercialization Professionals. Dr. Michael Lewis, a computer scientist and cybersecurity specialist, Vice Dean of School of Science and Technology, Nazarbayev University. So, rules are explained. We met our judges. we have all our 9 contestants here, and we've locked the doors. Can we lock the doors? It's time to bring our science stars. So please support them by being loud and laughing at their jokes. Okay, so first we have speaker from Spain, Alba Aguion. Alba is the winner of Fame Lab Spain 2016 and runner-up of Fame Lab International 2016. During her master's degree at the University of York, UK, Alba investigated one of her passions as biologist, ants. Now, as a first-year PhD student, she has moved away from insects to study shellfish. Alba will reveal to us the fascinating world of ants, introducing the beautiful concept of swarm intelligence. She will explain how these tiny creatures navigate over long distances, find food, and care for their young with a tiny brain. So, swarm intelligence, what ants can teach us. Please, Alba, floor is yours.
Thank you. What a big audience, and how well organized. Each one in your own seat, working as a team. Very nice. Do you know what you're reminding me of? The ants in my lab. My master thesis was all about them, and I'm here today to tell you about their organization and intelligence. It's fascinating how ants, with such a tiny brain, are able to organize themselves into perfect rows without following any instructions. Way better than you, who usually require a numerated ticket to organize yourselves in a theater. Ants, however, they follow no rules and they have no leaders. A colony of millions of ants functions just fine with no management at all. The queen ant does not tell the other ants what they need to do. The queen ant, she has what it takes. She's all ovaries, and she dedicates her life to laying eggs. So, if ants are not that intelligent and no one tells them what to do, how do they organize themselves in such an efficient way? By using pheromones to communicate by smell. If an ant finds a dead worm, she will go back to the nest laying a trail of pheromone that other ants can smell and can follow. Is the axe effect of ants. Well, for the British, it will be the lynx effect of ants. If more than one ant finds the dead worm and they use different paths to get to it, the ant that uses the shortest path will be able to make more trips and lay more pheromone. So they will attract all the other ants to that path. So while searching for food, no ant sees the big picture. But thanks to the communication between all of them, an intelligent behavior appears to know which route is the shortest path to get food. This, in science, is known as swarm intelligence. Many individuals with no central control that organize themselves to produce a complex behavior. Ants have been practicing swarm intelligence for millions of years, something that is really important nowadays in science for the construction of robots or for a further understanding of how neurons organize themselves in the brain. So if you go to the theater, don't lose your tickets, because you need them to know how to get back to your seats. But next time you step on an ant, remember all the things they have taught us. If you walk alone, you might get to your destination a little bit faster. But with company and communication, you will get much farther. Thank you. Thank you.
Thank you very much, Alba. So now we have 2 minutes of questions. Judges, please. Do you have questions?
You mentioned thinking in small brains, but is there any real thinking going on here?
Yeah, it's not really thinking. Yeah, that's true. It's not really thinking. Ants are very, very simple, and they follow really basic rules. So I don't think it's really thinking. The intelligence appears because of the communication, but the communication between all of these simple things, intelligence appears, but it's not really thinking like us. Yeah.
My question is, so in your opinion, what do you think, what we can learn from ants as human beings?
As human beings. Okay, so ants, they follow, they organize themselves very different than us, because like I said, there are no leaders, there are no rules, so it's a very different system that we have. And right now in science, we're using that system, for example, to construct robots, because it has been shown that instead of having one very big robot that is really powerful, it's better to have very small robots that are very, very simple, but that they are able to communicate with each other. Because if you have 1,000 of robots, if one of them fails, the system can still work. If you only have one robot that is very complex, but one piece fails, then it cannot work. So we are learning, that kind of cooperation and communication. We are also getting to understand a little bit better how the brain works, because the brain is also made of neurons that are really, really simple, and intelligence appears when all those neurons communicate with each other. So all these kind of systems, we can get a better understanding when we study how the ants communicate with each other. Thank you.
Thank you very much for this fascinating speech.
Thank you.
Thank you. So now we have our second speaker. She's from Kazakhstan, Dana Akılbekova. She is the winner of the first Fame Lab Kazakhstan in 2015 and first ever Kazakhstani participant of Fame Lab International. She has a bachelor degree in physics from Al-Farabi Kazakh National University. She holds a master degree in nanophysics from Rice University and PhD in material science and engineering from Iowa University. Her research interests lie in the fields of biophysics, biomedicine, and tissue engineering. Today, she will talk about green world and how bacteria can make our world green. So please welcome Dana Akılbekova.
Attention! Microbes! They're everywhere! Run as fast as you can! Wash your hand as hard as you can! That's what my mom used to tell me before the dinner. In my mind, I had a terrible image of these disgusting and these terrible creatures that trying to infect and kill me. Later, I learned that there are the only few species that are actually dangerous for us. Most of them are good for our health and for the environment. Without them, the life on Earth wouldn't be impossible. They hold the key to solve the problem of global warming, cleaning the environment, and even creating the cure for the cancer. See, I told you, Mom, I don't have to wash my hands again. So what's special about these microbes? Well, we live on Earth, remember, right? It's a closed system. All the elements that we need for our life, we get from the crust of the Earth that were formed there billion years ago. These elements are the sulfur, nitrogen, carbons, and other. They cycle through the ecosystem, moving from living to non-living forms and the locations. And the microbes plays important role in these cycles, especially in the nitrogen cycle. What is a nitrogen? It's a chemical element with a symbol N, with an atomic number 7. And it's a building block of life. Your genes, DNA, RNA, proteins, everything made of the nitrogen. But animals and plants, they cannot just get the nitrogen from the atmosphere like engulfing it, right? So that's where microbes come to play. They're able to decompose the nitrogen from the atmosphere into the different forms of the nitrogen, so the plants can consume it, and then the animals can consume these plants. These microbes, they have a special enzyme. They can decompose it, this element, called nitrogenase. What happens when your animal dies? This animal and these flowers, they die. what happens, there is another type of bacteria and microbes that are able to decompose this dead corpse and bring back the nitrogen to the atmosphere. So the cycle is complete. And these cycles keep going, and that's what makes our lives possible. Scientists were inspired by these amazing abilities of microbes to change from one form to another. And where we can use it? We can use it to clean the water from the oil spills. There is a special type of microbes that are able to decompose the oil into something harmless. They eat the oil, so we can just feed them oil and go on, yeah. But the problem again, people, because people create far more pollution that the microorganism can keep up with our pace. We are very fast. So what we can do? We can create genetically engineered bacteria. We can give them more nutrition, more food, so they can work faster and more efficient. What's the amazing thing about the microbes? They can decompose everything. They can decompose heavy toxic metals, plastic, paper into the car fuel, and the human waste into the rocket fuel. Ultimately, it's only microbes who can remove the oil spills from the ocean. In the long run, it's only microorganisms who can clean the environment for us. They're smart, they're tough. They created our world. Now, the microbes have to save us again.
Questions?
Aren't you concerned about runaway microbes? If they begin eating the oil, maybe they eat all the oil.
Well, that's actually, this is one of the problems that why a lot of oil companies don't use the microbes, because they actually degrade in the deep water, they degrade the pipeline. So they actually create the oil spills. So it's like weird. They create the oil spills to eat this oil, and it's an endless cycle. That's why we have to be careful how to use them. And they're living beings. You cannot just control them easily. They're like teenagers. They can be crazy. They can go any way they want. So we have to be really careful with them.
Actually, what do you think is the future for microbes?
The future? Well, I can talk from the brothers of my population of the microbes that they have a very bright future. Well, for sure, when we were gonna all die, they would try to decompose us and maybe create something better than humans. So that's a hope for them.
You named so many different applications of microbes, but in your opinion, what is the most important for people?
For people, it's cleaning up the Well, the one that actually more applicable that we can actually do is create some cure for the cancer. So, because you don't need a lot of them, you can control them, and it's in human. We have a lot of humans. We can try anything. So I think it's something that we can for sure try and see what's happening. It's already going, and it can be easily manipulated. Yeah.
Thank you, Dan. It's so informative and so funny as well. So thank you very much for your speech. So we have a next contestant, Diaz Argandikov, from Nazarbayev University, runner-up of FameLab Kazakhstan in 2017, which was just this March. He is a medical student and previously obtained his bachelor degree in biomedical sciences. His dream is to become a great doctor who will be able to make a difference in people's life. Tonight, he will talk about the nature of pain. So please welcome Diaz.
I think it was a mistake, but that's okay. Yeah, I think it was a mistake in the topic because I'm going to be talking still about my star zebrafish that I'm working on. Yeah. So.
Thank you for correcting.
That's okay, no worries. Yeah. Can I start? Yeah. Okay. Good evening, ladies and gentlemen. If you had to guess what animals scientists use today to test their research hypothesis, for example, the harmful effects of the environmental pollution on our health, you would probably think of rats or mice, but not a fish. So today, I would like to introduce you the star of my research, a zebrafish, which is best known as an aquarium pet that has recently become one of scientists' best allies in fighting against devastating diseases, among which, for example, are birth defects that might develop as a result of environmental pollution. But what is so special about this little fish, and what are the advantages of using it in science comparing to rats or mice? Well, first, it is simply a matter of numbers. At one time, one zebrafish can produce up to 200 eggs. Plus, it's almost 90% cheaper to maintain them when you compare them to rats or mice. Also, they grow extremely fast, developing as much in one day as a human embryo develops in 1 month. But what is more, zebrafish embryos are completely transparent, which make them remarkable to study, as you can actually see what's happening inside their bodies without touching them. And that is not something you can do with other laboratory animals. Finally, and most importantly, scientists now have a complete map of genetic information of zebrafish. And using zebrafish, scientists today study genetic disorders, skin cancers, blood diseases, and birth defects that, according to research, might be caused by environmental factors, such as pollution. For studying these effects of air pollutants or toxic metals that might be present in soil or in water we drink, zebrafish embryos are becoming a very great model and unique. And because of their uniqueness, 18 zebrafish have been sent to the space station for studying, guess what? Muscle atrophy, or the loss of muscles that can occur under the condition of microgravity. Is it amazing that this little fish is now being used for studying the things we couldn't even imagine many years ago? To conclude, there is no ideal animal model for studying human diseases because our body is so complex and sophisticated. But now you know why this little fish has become a scientific star, not only of my research, but in the world of science. And next time, when you see zebrafish in somebody's home aquarium, I'm sure you will be able to appreciate a remarkable scientific importance of zebrafish. Thank you.
Are there other fish that compare? Do they come anywhere close?
Yeah, actually, so there's species, there are actually species, they call, the Latin name is Danio revia. So in Russian, when you go to the pet store and then you say zebrafish, nobody would understand you. But you say Danio revia, then everyone understands you. So within this class of the species, there are multiple others, but that one's Danio revia is the best ones to study, and they've been, they've been involved in the science projects for many years already, and they attract lots of money for their grants because they produce really reliable results. Yeah. Questions?
Any more questions?
Can you tell us the story behind zebrafish? So, like, who was for the first time using it? When the articles and research papers started?
Yeah, that's a very good question. I think it was developed many years ago. But if I say that, I won't be too specific. But I think it was in early 60s or late 70s. Yeah, and so they identified this zebrafish. They did a couple of experiments, and they saw that they really produce similar results. At that time, the only animal models that have been in use are, of course, mice, rats, and also chimpanzee, but they're all expensive to maintain. So, also doing an experiment, you have to have numbers to have a statistical significance. But using zebrafish, you can have them a lot, you can maintain them, you can keep the experiments under control, and then produce a lot more results that would be reliable. But of course, that's not the end of the story, because if you test one drug on the zebrafish and then you sell it to the people, that doesn't work. You have to go from zebrafish to rats to or mice, and then chimpanzee, and then human clinical trials. But as a first point, zebrafish is a better option to use, as the same as mice or rats to a certain extent. Thank you.
Thank you very much. We learned a lot about zebrafish. Next speaker is from Norway. She is the winner of Fame Lab CERN 2015 and runner-up of Fame Lab International 2015. Lilian Smestad. She obtained her PhD involved in the experiments at the Large Hadron Collider, a particle accelerator at CERN, one of the world's largest and most respected centers for scientific research in Switzerland. She studies the smallest building blocks of nature, and she will talk about antimatter today. For some of us, it sounds like a science fiction, but Lilian will show us that this antimatter is actually a science fact. So please welcome Dr. Lilian Smestad.
What's the matter with antimatter? In fact, we will have to start somewhere else. What is antimatter? Don't be fooled by the name. Antimatter is perfectly fine matter. You could build something out of it if there were sufficient amounts of it around. It's anti in the sense of opposite, ordinary matter. And it's indeed when the two of them meet that things get interesting. It's the relationship between matter and antimatter which is of the essence for this story. The leader of my experiment likes to call their encounter a tango of death. They attract each other and they destroy each other in a, relatively speaking, extremely violent explosion in a flash of light. Afterwards, they're both gone. Left is pure energy. Well, this is certainly a peculiar relationship, but does it answer our initial question? What's the matter with antimatter? When it comes to antimatter, the real question is as fundamental, as existential as this. Why are we here? Because in moments, in the Big Bang, moments after it, when everything we know exploded out of a hot and dense state, we believed that there were equal amounts of matter and antimatter. And so all of that opposite matter met, engaged in this deadly tango, and left was only energy. But we are here. I'm standing in front of you, asking this question, and how can I? I am made of matter. There must have been the slightest of imbalance between matter and antimatter in the early universe, such that when everything was destroyed, there was something left. Personally, I'm very grateful for this imbalance. But what caused it? And the truth is, we don't know. For all the things that we know about particles, and we've learned a lot about particles the last 100 years, we don't know the full story of how matter and antimatter gets treated differently. And our approach for finding out is by studying antimatter to the greatest details we possibly can, to search for differences in its behavior and characteristics with respect to the one of ordinary matter. And this is our hunt for a fundamental physical explanation for our existence. And that's why finding out what's the matter with antimatter matters. Thank you.
Thank you very much. Questions from our judges?
When you phrase the title, "What's the matter with antimatter?" it suggests that maybe there's something wrong with antimatter. Is there anything wrong with antimatter?
Oh, I love that question. Well, it depends. You know, in physics, okay, we love this sentence. I have to say it. It's all relative. So maybe there's something wrong with matter and not with antimatter. The point is that indeed they get treated differently, slightly. And this was a shock when it was first discovered in the mid-1960s, that processes involving matter and processes involving antimatter, they move at slightly different speeds. And this could be a reason for why matter won over antimatter in the early universe, and thus the reason why we're here. And we have some examples of such processes, but they're not strong enough to explain the observable universe as we see it. So we have to look further for what's wrong with matter, antimatter. Sorry, okay. Yeah. Still.
Thank you very much. Next speaker is Tomiris Atajanova from Kazakhstan. She is one of the brightest participants of FameLab Kazakhstan 2017. She is from Almaty and a graduate of Nazarbayev University in Biological Sciences. Currently, she is studying in medical school and just completed her first year. Tomiris was always interested in communication and science, and she has a minor in communication as well. She will talk about men's nipples and why they have them at all. So please welcome Tomiris.
Imagine the human body. Imagine the male human body. Try to focus on his breasts and nipples. Wait, what are the nipples doing there? Have you ever thought about that? Well, I didn't have an idea until one of my friends asked me the striking question. Hey, do you know why do men have nipples? Today, I will reveal you a secret. All we know that for mammals it is crucially important to breastfeed our newborns. Thus, the maternal immunity is transferred to them and is helping newborns to defeat infections. But you have probably noticed that fathers don't do breastfeeding, right? Well, at least mine didn't. So, what makes our bodies look so different? The answer is our genes. People usually have 46 chromosomes, genetic material, in each body cell. 2 of them, known as X and Y, are called sex chromosomes, and they determine whether a person will develop male or female sex characteristics. Thus, mothers always pass X to their offspring, whereas fathers can pass either X or Y chromosome. It means that father's genetic material determines the future sex of the baby. In the womb of the mother, the baby who inherits the X and X combination of genetic material will develop into a girl with beautiful hair like mine. The baby who inherits X and Y combination of genetic material will be developed into a masculine boy, like Diaz. So the interesting fact is that during the first 6 weeks of the development, the baby has potentially 2 genders until the SRY gene of Y chromosome, sex-determinant region of Y chromosome, kicks into play. It produces a hormone, testosterone, which destroys all of the future female reproductive organs. However, by that time, the breast tissue has been already formed. And since it doesn't cause any harm to male body, it just stays there and is not eliminated. But what if this SRY gene doesn't work and testosterone is not produced? The baby, by default, will be developed into a girl, even in the presence of Y chromosome. The condition is known as Swyer syndrome. Now you know more about beautiful accessories on your chest. And we all need to realize that if it wasn't for testosterone, I would probably talk to an audience consisting purely of females. Thank you.
Now I'm a little bit worried. Do we have any other legacy attributes that maybe we shouldn't have?
Well, why shouldn't? It's just, it's not necessary that you shouldn't have them, but what makes the person in the beginning of the development, like, it means that we are all kind of created phenotypically from the one creature, I would say. So it doesn't mean that you necessarily, it wouldn't be fun if you wouldn't have nipples on your chest, you know.
This is a very interesting topic, delicate one. So my wife every time complains that, you see, you are a male and you don't feed a child. So is it possible in medicine to make men to have this function?
Yeah, very good question. Thank you. So, if actually males, like men, they have the breast tissue that has been already formed. So even if you don't see the actual breast, you have some tissue that can be developed to produce a mammary gland. So if you have a hormone called prolactin, and if the sizes of it, well, the amounts of it increases in your body, or if you inject it externally, actually men can do lactate. So, surprise, right?
Thank you very much. So many questions from our male juries. Our next international guest is Emer Maguire from Northern Ireland. Emer Maguire is the 2015 winner of FameLab Northern Ireland and FameLab UK. She is a speech and language therapist with a master's degree in clinical anatomy and likes to combine her love of communication and science through science communication. Since winning FameLab, Emer has gone on to deliver talks at TEDx and to promote science to the general public on radio and through science festival events all over the UK and Ireland. Emer is currently working with BBC Radio Ulster presenting a show on science and tech. It will be aired this summer. She will talk about the science behind fascinating topics such as love, communication, and flirting. So please welcome Emer.
Thank you. I got hooked on a drug once. It created an altered chemical state in my brain that I haven't recovered from since. It made me do crazy things, but I had no control. I was addicted. My drug, love. As a recovering addict, I now know there are 3 distinct stages of love. So join me on my journey of madness. We begin at stage 1, lust. At puberty, 2 sex hormones become active in the body, estrogen and testosterone. And from then on in, we are constantly on the prowl for someone to reproduce with. How do we entice them? We flirt. Some of us better than others. Flirting ignites an animalistic part of the human brain, giving us 2 choices: approach or retreat. Obviously, you all like what you see, and I watch as you come towards me. Come with me to stage 2, romantic attraction. People associate love with the heart, but the real magic happens in the brain. And using an MRI scanner, scientists were able to see the parts of the brain that light up when people are madly in love. And I happen to have the results right here. The first, thank you. The first is the caudate nucleus. Ding. This helps us expect and detect rewards. In this case, love. The second is the ventral tegmental area. It is a chemical making factory. Bing! Our own personal Cupid shooting arrows laced with love drugs into our vulnerable brains. These love drugs are serotonin blockers, adrenaline and dopamine. And this chemical cocktail gives us a natural high. It stimulates the same area of the brain as cocaine with similar side effects, like increased heart rate, obsessive thoughts, and ultimately, addiction. Put simply, we are chemically insane. However, our brain can't survive in that drugged up state forever. So it sobers up, guiding us to stage 3, attachment. You are now in this for the long haul. Your brain responds with a rush of oxytocin, the love hormone. It is the glue in a long-term relationship, keeping us together long enough to raise a family. And I suspect many of you here tonight will fall victim to this drug called love. with me. So next time you find yourself doing something crazy in the name of love, don't blame yourself, blame science. Thank you.
Thank you very much.
Thank you. Thank you for a wonderful presentation. So there are different types of love. Love for parents, love for children, love for your beloved one. What is the science behind this? Is there any science behind the types of love?
Yep, there is. So there's, as you said, love between a parent and a child, or between two parents together, which is a very different type of love. And it's all to do with chemicals. So as I said there about the chemical cocktail with things like dopamine and adrenaline, those are really feel-good hormones. And that's the kind of love you feel with someone that you're going out with, or someone that you're obsessed with, or a celebrity that you're in love with. That's the type of love. But the type of love that is felt between kind of a mother and a child, or a father and a child, that is a more bonding love. And the thing beside that, the thing behind that is a chemical called oxytocin. You have oxytocin in, love between partners as well, but you also need those other things. So oxytocin in terms of family is more of a bonding experience, and it can be released when you touch someone or whenever you hug someone, which is why parents should be really cuddly with their kids. It's also something that is released whenever a mum breastfeeds a child, or whenever a parent has skin-to-skin contact. That's why now in a lot of hospitals, as soon as a child is born, they're giving the child to the parents with their shirts off so that they can have skin-to-skin, so they can have this hormone. And as I said there, the oxytocin hormone is called the love hormone, but because of its impact in kind of sibling relationships and parent-child relationships, some people also call it the cuddle hormone. Thank you.
Is there any other feelings that can have the same effect to our brain as love?
Is there any other feelings that can have the same effect as love? Yeah. Do you want to know that for a particular reason, or? So one of the coolest things that I think can have the same feeling as love, the main ingredient I think with love is dopamine. So dopamine's a really feel-good hormone and chemical, and it really lights up the pleasure centers. These are all the pleasure centers in your brain. But you can light these up from some other things, and the best way to do it is by doing novel or interesting things that you wouldn't usually do. Like coming to Kazakhstan, this isn't very novel for me, so my receptors are going off. But one of the best things that you can do is jump out of an airplane. So either get a boyfriend or jump out of an airplane. Thank you.
Thank you, Emma.
Thank you very much.
Thank you very much. Love is in the air. So our next speaker, also from Kazakhstan, Damel Mektibayeva. She is a biotechnologist and innovator. She got her bachelor degree in biotechnology and chemistry at Indiana University and master in biotechnology at École Supérieure in France. She is a winner of the Global Impact competition held by Singularity University at NASA Center. Her project, Medical Device for Fast and Painless Detection of Infectious Diseases, won a number of international competitions, such as Health Tech Track at World Cup Tech Challenge in California, USA, Health Track at Hello Tomorrow Challenge Paris, France, Startup Chile, Startup Peru, Google DCOM Prize at World Startup Nations, Seoul, South Korea, and Startup Mexico. Now Damel is a mentor at several startups in biomedicine area and the senior manager at the Innovations Development Department of Nazarbayev University. Damel dreams to translate her ideas into healthcare, namely diagnostic systems that will be available to people in any pharmacy store. Have you ever heard? So today she will talk about racism in cells. Have you ever heard of racism in cells? So Damel will tell us about this.
Many people think that cancer is an invader, like a bacteria or a virus, and do not realize that cancer cells are our own cells, which suddenly go wild. Despite its microscopic size, every human cell is a universe with hundreds of processes going on inside of it. And every action in the cell is very highly regulated, like in a very well-organized country, let's say a Scandinavian one. And like in steady Norway appears Breivik, cancer cells appear from normal cells. Cancer cells act like racist group. They violate the laws of the country. They do not listen to the external signals to stop growing and divide. They lose family ties and, like lonely anarchists, detach and migrate to other organs. There, they recruit other normal cells to share their values and views. Finally, in the end, they grow into a greedy accumulation of cells, which grow uncontrollably and fast. While forming a tumor, they also grow new vessels to extract all the nutrients out of the body. These crazy cells think that they deserve more than others. But who usually stops racism in the country? Yes, it's police and intelligence services. And you know what? There is good news. We have an analog of MI6 in our body. That's a protein P53. P53 constantly checks the cells and prevents them from becoming cancerous. And that's actually what we do at Nazarbayev University. We have a project where we use chicken embryo extract to activate defense systems of an organism, to activate P53 and other proteins, and make all the processes go back to normal again. So potentially such project can become an alternative treatment for onco patients. Actions of cancer cells, like actions of any racist group, is illogical and meaningless because in the end it results in death. State dies and tumor dies, valor margulis as they say. But as any form of racism, cancer should be stopped. Thank you. Oops. Too fast.
You compared the body maybe to countries. What would be the right balance of, what would be a good central country, a good model for you between one extreme and the other?
Yeah, I compared because actually, as I said, every cell is like the whole state because there are like hundreds, thousands of things going on. But I just wanted to say that even healthy people can get cancer because, like steady Norway, you know, because it doesn't affect only weak people. It can affect any people just because any normal cell can potentially become cancer cell. But as a racist group, they can be stopped.
Thank you very much, Damir. One more applaud. So next runner is Artur Saudabayev, a winner of Fame Lab Kazakhstan 2017. He was born in Shymkent. He achieved a lot in his life. At different times he were awarded Altın Bilgi, Bolashak, Talab, British Gas Kazakhstan scholarships. He is now in the Department of Robotics at Nazarbayev University. Artur strives teaching science and technology classes with emphasis on the future implications on life of people and society in general. He will talk about virtual reality, bringing the art of storytelling to a whole new level, and how we can leverage this emerging immersive technology to improve people's lives. So please welcome Artur Saudabayev.
I just wanted also to say that the topic is different as we had. I'm going to talk about the network-based approach for identifying protein related with diseases. If it's okay, sorry about that.
Sorry. I must have been updated. Sorry.
Everything in this world is connected, and every event is the result of a certain process. Our DNA contains genes which produce proteins, and proteins are essential units which enable all biological functions in our bodies. They transport oxygen, they attack disease cells, they even digest food for us. Now, it is critical to understand that proteins don't work in isolation. Any biological function is the result of an intricate common effort. For example, if you drink milk, first type of protein detects lactose in your system. It then sends a signal via a messenger protein to cell nucleus, where third type of protein goes to DNA and tells that it needs to produce, finally, fourth type of protein, which is called lactase enzyme, which is used to digest lactose. There are multiple types of relations which can occur between proteins, and it can be very well modeled as a network, where node is a protein and edge is a relation. And this digesting process will be only a tiny cluster within a vast network of thousands of proteins and millions of connections. Now, scientists identified that lactose intolerance happens due to lack of lactase enzyme. But we now know, you and I, that it's also maybe because of either of these three, or maybe all of these three proteins, that we fail to produce enough of lactase. And similarly, for other diseases, scientists can identify the original dysfunctional protein, tag it in the network, and run statistical tests to find out if other molecules are related and potentially connected to the condition. They become suspects, and they're taken to the lab for further investigation. Now, this technology enables rapid discovery of genetic basis for human disease, and its implications go far beyond healthcare. 2 years ago, I took a plastic tube, I spit into it, and posted it. Oh, for a genetic test, of course. A few weeks ago, I obtained my report stating my chance of developing Alzheimer's, a disease which leads to loss of memory, social withdrawal, and eventually death. Now, it struck me, this knowledge can completely change my life because I'm also part of the system, I'm also communicating with other people. And what if I do have high chance of Alzheimer's? And what if other people will find out? Shall my insurance company charge me more than other people? Or shall no one gonna marry me because they don't want this gene set in their kids? Maybe my employer will not promote me, well, because my brain might let me down someday, right? Will my gene set eventually be the reason for loneliness? Thank you.
With your models, how abstract or how detailed could they be to be useful for you? How much detail is necessary?
The network models. It can go at very different levels. The thing is, the complexity of disease and all the involved parties are so high that you need to do the systematic approach and study them at different levels. For example, they can be physical or chemical connections or common function like here. Sometimes we just care that there is a fact of interaction, binary, yes or no. And we do the link, and then we draw our network and perform some graph theory analysis to identify related nodes. Sometimes you want to look exactly at something, if it's a chemical connection, and then you do extrapolation based on that. So it can be both, but it is quite precise, because we do have exact node proteins, and we do have exact links and edges as interactions.
Thank you very much. One more question.
In your opinion, do you think that in the future there will be some time when all diseases, all illnesses will be cured, or will find any remedy for all of them?
Well, in the long run, I'm quite optimistic. I do strongly believe that we will be able to cure or prevent most of the chronic diseases we know today, which cause a lot of problem and troubles and tragic to us. But maybe we'll also develop new ones, because they all evolve, the mechanisms evolve, so we need to adapt on a constant basis with time. But there is all the technology, all the science, and the dynamics which shows that we should be able to do that indeed. Thank you.
And our last but not least speaker the winner of International Fame Lab in 2016. He is a genetist at the University Putra, Malaysia, and obtained his doctorate in oncology and genetics from the University of Cambridge. He chairs the Young Scientist Network Academy of Sciences, Malaysia, that represents the top young Malaysian scientists across all scientific disciplines. He was appointed as a member of the Global Young Academy. He is a big proponent of science communication and science leadership, and conducts various seminars and workshops to promote these areas of science in the region. So please welcome Abi Veer Kumarasivam.
Hi. Good try.
I was practicing, but sorry.
Salam Kazakhstan. Malaysia, Kazakhstani, Jaksi Koredi. It has been an amazing 2 days in Astana, and stunning physical infrastructure, rich culture, great university, and more importantly, beautiful people. And it's really, really evident that life is beautiful. And life starts when two cells come together, and these cells grow, and they grow into the trillions of beautiful cells that we are. But what controls this growth in a precise manner is the superpowers of knowledge, our genes, and we have heard a lot of them today. And we have 25,000 genes, thereabouts, that are essentially recipes to make the millions of proteins that shape our body. And it's these that also makes us very unique. But how long do you think is the DNA in our cell? Well, it's 1.8 meters tall. And it's pretty amazing considering how small the cell is. And help you to visualize, if the cell was a ping pong ball, you would have to go up to the moon and back to see the stretch of a DNA. And today, I went to the really impressive Baitarak Tower, and if you ran up and down 4 million times, that would be the length of the DNA. Amazing, isn't it? And we all have essentially the same 25,000 genes, but the exact content of that recipe that makes you the beautiful and unique individuals, and that is the other superpower of the genes. Just like how we all have bags, and essentially the function is to carry stuff, but some have Prada and some have Gucci. And that's essentially the difference. And that's what makes us really, really, really good. But the difference between you, and yourself, is essentially 15 million differences in the DNA. And it's because of this complexity, as Arthur had talked about, makes it difficult for scientists to identify what makes the amazing Olga Ripakova jump, Usain Bolt running, or Einstein's brain, or Kim Kardashian's talent, I suppose. Unfortunately, with greater power, there's greater responsibility. And this is where we realize now that the environment can actually affect and change the content of our genes. And this is what we call mutations. For a single cell to become a cancer cell, it acquires multiples of mutations in these genes. But wait a minute, without any superpower story, we have to expose some sexy body parts. And this time around, the organs are in the right place. Cancers hijack the genetic mutations, and they hijack the ability to now have new superpowers. One example of a superpower is to be able to invade and spread to other parts of the body in a process called metastasis. And this is where, for example, a cancer that was originally in the intestine now migrates into the liver and into the lung. And in the last 5 seconds, 2 individuals have found out they have cancer. 1 life has been lost because of cancer. And this is our battle against cancer. And this is why we are so keen about genetics, and you've heard so much about that, because scientists have and are continuing to identify the genetic mutations and superpowers that are hijacked by cancer cells, and then to work on trying to identify how can we overturn this and identify these particular genetic mutations and switch them off and target them, neutralize them, and kill them. Although genetics is going to revolutionize the way we treat patients in the age of precision medicine, the reality is that our genes do not control our destiny. In fact, our lifestyle, our choices of lifestyle ultimately controls the way our genes behave. And this is why effective science communication is extremely important, because it not only improves public awareness, but can also impact political will. Gelimin ote manisde, Gelimin tamsanu ocean, galim bolu kajet yemes. Rahmat. Sure.
Can you please reflect on the ethical side, working with DNA and working with all these mutations?
Absolutely. There are multiple areas in this. The first is, of course, the information that it's going to be identified, and if we are based and we are judged based on our own genetic makeup. One of the biggest problems, which I think Arthur mentioned, is penetrance. And I tried to allude to the fact that genes alone do not determine fate. There are multiple factors that actually affect. In fact, a fetus in the stomach, in the uterus, will also already be determined to a certain extent, many attributes, by the behavior of the mother alone. And so the first problem is the confidentiality of the information and how this information is going to be hijacked by insurance companies, and how is that going to affect an individual's predetermined future per se. The second issue, and probably a bigger problem, is how do we change our ways in identifying individuals, treating individuals. For example, the reason why the world is so unique is because all of us are different, and it's that diversity that we actually celebrate. I think if everyone could jump like Olga, we won't be celebrating the London Olympics when she won the gold medal, right? I mean, all of us will be, saying, that's nothing special. And an issue such as CRISPR, the new technology in where we can actually genetically edit, I think the problem is how do we make sure that this technology is not edited, and we have had, it's not too far away, well, it's only in the 1940s, 50s, and 60s where some really crazy experiments were actually done, that we need to be aware that this technology, because it's so powerful and so precise, it can be hijacked. Yeah.
You mentioned briefly insurance companies maybe
Sure.
having this information. Do you think insurance companies should have genomic information of individuals?
Well, speaking on behalf of the society, I think we shouldn't, especially not now, because for the majority of genes, even for highly penetrant genes, where if you know a particular mutation and you know that's going to be a fate, I think it's really unfair, because there is still an element of probability and risk, and I think that I would really be petrified or even devastated that if I tested my son and I found something, and that's going to determine, that stigma is going to be there. I feel that we have to view insurance companies as a business, and why should it be that the public, the society, the average man is the one that actually has to take the risk of that probability, when it should be the insurance that also has a role in that. And I believe that they shouldn't. Because one problem that we have in Malaysia, where we run a genetic test for individuals, because we have a problem where kids drop dead in the fields, while playing football. And it's a huge problem for young onset of heart attacks. And we have a genetic test that we can actually test it. And we have families where we think that they should test, and the only reason they don't want to test is because this would mean that the kid will no longer have the same kind of support and care. And like I said, there's still an element of probability, and that shouldn't be determining how the individual lives. Yeah, thank you. Thank you.
Thank you very much. So this was the final speech, and thank you very much to our participants, to our speakers, for your fascinating speeches. You're all phenomenal. You are really true science stars. However, it's a competition, and the judges are going to decide on the winners. So they are on their way to decide, to consult and decide who is the winner. And while they are discussing who wins this competition, we have some some work to do. So let's play some games and let's make our brains to work. The rules are quite simple. We need 6 volunteers. So we need 6 volunteers. Who is going to be a volunteer? Come on, just 6 people, 5 more. Yeah, so we have 3 volunteers, 3 more volunteers. Okay, so 1, 2, 3, 4, 5, and 1 more. Come on, don't be shy. Okay, so you will be shown with different words, and other people won't see it, only you, okay? Only you and me will see what's the word. And what you need to do is to explain it to the rest by using only your body language and gestures. So no words, and don't try to show like L, M, whatever, okay? So just your body language and gestures, okay? So let's wait, where are our words? Okay, so here they are. And you have just 1 minute to show, okay? And yes, you can skip some word, and those who showed, and not only showed, but also if people guessed what you're showing. So if you have more words, then you are the winner, okay? So shall we start? Okay, so you'll see it from, who is the first one? The beginning, okay? So 1 minute, and let's count how many words you can explain, okay? No, no. Close, close. Inertia, okay, so next one. Compass, okay, correct, next one. So it's close to microbe. Yeah, bacteria, okay. No, I don't have timer. А кто время смотрит? Okay. Okay, 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 okay. So, what's your? Do you have a watch? Can we find one timekeeper? I thought Agirim is our timekeeper, but okay. Thank you very much. So now we have a timekeeper, but you already have 4 words, right, guest? So it was a practice, and if someone else. Okay, so let's start from the beginning. Who is going to start? Who wants to start? Okay, so what's your name?
It's my Alexander.
Okay, Alexander will start. Rocket. close to people. Population. Close to contamination. Okay, we skipped it. Flower. Stop, so you have 3 words. So applause to Alexander. What's your name?
Dana.
So, okay, Dana, let's start. Okay, skipped. Meteor, meteor. Shadow, correct. Heat. Magnet. Atom. So you have 5 words. Dana has 5 words. What's your name?
Jamila.
Okay, Jamila. Let's start. Map, okay. Lan, okay, lan is correct. Skip it. Skipped. Next one. Exercise. Nope. Okay, somebody said insect. Yeah, she's done it. It's time? So you have 4 words. What's your name?
I'm your namesake, Asia.
Oh, okay. Asiya. Easy to remember? Tornado. Mass. Yeah, somebody said mass. Nope. Graph, yes. Okay, you have 3 words, Asiya. What's your name?
My name is Hulk.
Hulk. Oh, you have Hulk here. Star. Skin. Heart. Wow, this is so easy. All medicine. Organ. Molecule. Virus. Yes, so 6. Space. Predator. Yes. Predator. Predator. Stop. Oh, you have 8 words. Wow. Hulk. So what's your name?
Ruslan.
Okay, Ruslan. Do we still have cards?
The ones that were not used.
No, he didn't start. I mean, he was the one who started again and again, but he didn't run. He didn't compete, actually. He didn't give a chance yet. Yeah. Antenna, yes. Yes. Gravity. Yeah, someone said gravity. Friction. Doctor, okay. DNA, yes. 6. Ocean, yes. Oh, so you have 7 words. So we have Hulk with 8 words, Jasolan with 7 words. Do you want to compete? Because it's just one word and he did it twice, so let's give another chance to Jasolan. Okay, so let's go. Who is going to be first?
Let's see the word.
Ice. Okay, someone told ice. Root. Charge. Newton. Solution. Somebody said solution. Meter. So you have 6 words. Earthquake. .3D printing. Calculator. Exam. Wow. Oh gosh. Oh, this is not fair on you. So it's 2 words, okay? Because it's the only one. Okay. Sorry, Jaslan. And the winner is Hulk. Oh, I'm trying to find this out. Do we have presents? Do we have? So let's wait.
There is prize, but unfortunately I didn't left, so he will be given the prize after.
So yes, please keep in touch. Stay somewhere around, okay? So we'll give you a prize. Ah, okay. So it's quicker than we thought. Второй раунд. So maybe someone else want to play, huh? Let's have another 6 volunteers. Thank you very much, guys. So we need 6 more volunteers. Who do you want to play? Come on, guys. It's fun. So it's the same game. Okay, so we need two more people. 2 more volunteers. Oh, don't be shy. Come on. Let's join. Is it okay? Okay, let's have 5. 5 is fine. So, shall we start? Come on, quick. So, this time we'll make it a little bit more complicated. You have 30 seconds, okay? So you have 30 seconds. And we'll count how many words you can explain in 30 seconds. Yes, you can skip. Yes, but just one skip, okay? So you need to think what you can skip. Okay, so let's start. Who is going to start? Come on. Ladies first, or maybe you'll be a gentleman and you start?
Okay.
What's your name?
Yedige.
So Yedige will start. Please come to the center.
But what if I don't know how to work?
If you don't know, just tell don't know, okay? We'll try to figure it out.
Okay.
Wind. Water. Skeleton, yes. Frankenstein. Okay, so you have 4 words. Okay, so what's your name?
Nazirke.
Nazirke, please start. Не видно руки. Skip. Bookworm. Google. Okay, so you have 2 words. Who's the next one?
Жамболат.
Okay, Jambolat, please start. So it's gas. Velocity. Okay, so you have 2 words. Who's the next one?
Aydın.
Aydın, please start. Protein. Stop, so you have 1 word.
Sabina.
Okay, Sabina, please come. Dinosaur. Pendulum. Volcano. Telescope. 4 words, wow. Okay.
Aygirim.
Okay, so Aygirim, please come. Professor. Erica. Gravitation. Mouse, yes. Laboratory mouse, yes. So you have 4 words. Okay, so we have 2 girls who guessed, who explained 4 words. So it's Aygirim and Sabina. Ah, you also. Oh, we have 3 people. Grand final. Okay, so who will start? No skipping. So who is the brave one? Do you want to start again? Don't let's make it tradition. Battery. Microscope. Laser. Einstein. Scientist. So you have 5 words. Wow. in 30 seconds. Aygirim. Oxygen. Wow. From the small. So the second one was force and now antenna. DNA. This is super quick. Star. Ocean. Magnet. So you have 7 words. Sabina, please. Magnet. Root. Root, yes. Electricity. So you have 3 words, and the winner is Aygirim. So please, Aygirim, this is your prize. Thank you very much, volunteers. And I've seen our judges back, right? So they might have a decision now.
Guys, it was a tremendous set of presentations, and judges always say this, but we had a very hard time coming to some conclusions. A lot of disputes, and you can see I'm a little bit damaged from the discussions. Because this isn't a normal competition, we decided to make up a little bit our own rules. We had a very hard time singling out just one presentation. So I hope you bear with us. We would like to distinguish 2 of the presentations. We're going to announce first a runner-up and then the grand champion, the prize. Okay? I think both of them are crowd favorites, so I'd like to offer up first a very provocative discussion, one that had us all captivated with no visual aids per se. We think that Tomaris did a remarkable job on the topic.
Tamiris Atajanova.
It was a great job. But next, we found the grand champion, the winner, remarkably evocative and mesmerizing, and somehow her approach worked on apparently all of the judges. Emer Maguire. Okay, thank you. And now I believe that there's recognition for the rest of our presenters this evening.
Diaz Argandikov. Lilian Smestad. Dana Akılbekova. Артур Саудабаев. Альба Агьон. Дамель Мектебаева. Аби Виера Кумара Сивам. are taking group photo. For coming and for being here. So that's the end. And see you later. See you next time.